Hello, my name is Tillie Morris, and through my research in the Weir Lab, which is in the Institute of Neuroscience here at UO, I've been studying the effect of optogenetic suppression of gap detection in mice. In the big picture, this research relates to speech processing, which is the part of hearing that turns the sounds you hear into electrical signals your brain can understand. Many people have deficits in speech processing. These deficits happen with normal aging and with diseases like Alzheimer's. One aspect contributing to speech processing deficits is having trouble with detecting the gaps in between words, syllables, and phonemes. As I'm talking right now, your brain is quickly detecting the brief gaps in between the words and syllables that I'm using, which allows you to understand what I'm saying. The ability to process brief gaps like this is known as gap detection. Since gaps play such a huge role in speech processing, the neuronal circuits used in gap detection are a simplified model of the neuronal circuits used in speech processing. So it's important to study gap detection. For this experiment, we were manipulating the gap detection ability of mice. We have already shown that mice can perform gap detection, meaning they can detect gaps in background sound. And now we're trying to manipulate that gap detection ability by targeting the activity of neurons in auditory cortex. If we can successfully manipulate this gap detection ability, then it will suggest that the neurons in auditory cortex are indeed responsible for gap detection. Understanding more about which neurons are used for gap detection will tell us more about the pathways used for speech processing, and understanding these pathways can help provide potential remedies for people with speech processing deficits. To accomplish this, we had mice perform a behavioral task called a choice task. This entails the mice going into a box that has three ports and a constant background white noise. You can see the setup for this choice task in the lower left corner. For each trial, there either is a gap in this background sound or there is no gap, and they have to respond based on what they hear. If there is a gap, they get rewarded by going to the left port, and if there is no gap, they get rewarded by going to the right port. The mice we use to do this choice task are optogenetic mice, which means they have genes that are influenced by light. These specific mice have inhibitory optogenetic genes, which means that the activity of their cells is turned off when the cells are exposed to light. The mice have fibers implanted in their brains that hover just over auditory cortex so that we can shine light on the neurons in auditory cortex. You can see this set up in the images here in the middle. In theory, since light inhibits neuronal activity of these mice, when the light is on, it should cause the mice to respond to the stimulus as if there is no gap, even when there is a gap. So this should make the mice's gap detection ability get worse. The data we got from this was inconclusive. As you can see in the graph, the mice went to the left port 40% of the time, regardless of the length of the gap they heard. This graph is only representing the data from one mouse in the experiment, but all the mice in the experiment showed the same strong bias of going to either the left port or the right port. For reference, for the longest gap duration, when it's the most obvious that there is a gap in the background sound, we expected the mice to go to the left port almost 100% of the time because they get rewarded at the left port when there is a gap. And for the shortest gap duration of 0 milliseconds, when it's most obvious that there is no gap, we expected to see the mice go to the left port almost 0% of the time. This strong bias that they showed indicates that they were not paying attention to the stimulus. In this case, the bias was due to a technical problem with the software that runs the behavioral task. We will need to redo these trials in order to get conclusive data. If we are able to manipulate gap detection and confirm the pathway, this part of the pathway for speech processing, this knowledge could have the potential to help improve the quality of life of people living with speech processing deficits.